again, Sandeep, for your very interesting talk, and thank you for being with us.
Yeah.
Let's move to the third speaker of this contributed session, who is Stefano Pagani. Stefano, can you hear me?
Yes, can you hear me?
Okay. Yes, can you share the screen with us? Let me introduce you Stefano Pagani, who is a researcher here at Politecnico di Milano. He's one of the members of the team iArt, and is going to give a talk regarding the numerical study of electrophysiological substrate triggering arrhythmias. Please, Stefano, the stage is yours.
Okay, so hello everybody. So this work is a joint collaboration between the iArt project and the Arrhythmia unit from San Raffaele Hospital in Milan. And so the main focus of this this talk is to try to okay, so it's to describe the process of choosing suitable model parameters in order to create mathematical models of cardiac electrophysiology able to reproduce arrhythmias. And so the process of constructing a model is quite complex. It's going from the definition of the input parameters to the numerical solution, and finally to the evaluation of outputs of clinical interest. Of course, we would like to build a model which is accurate and which also has some predictive power in terms of reproducing scenarios that are not measured when it's and are not used for its construction. Of course, there are a lot of questions about efficiency and about the choice of the model. We can rely on simplified model or on more or on detailed models such as the domain equation and so on. And so in today's talk, I will focus on the first part and on the last one, and in the framework of arrhythmias. So the anomalous rhythm of the heart could be generated in the atria or in the ventricular in the form of fibrillation or tachycardia. And the main ingredients of this process are, first of all, the presence of a trigger which starts the re-entry phenomenon. Then there is at the cellular level some modification of ionic currents which lead to head-tail interaction between the activation front. And there is also some structural defects in the form of fibrosis or in the form of scarring tissue. And of course, since the heart is a pump, we need to rely on the fact that there are also the hemodynamic and mechanical parts which are playing a role in the form of a feedback. So this is an example when you are dealing with, so in details about one of these pathology. And so in this talk, I will address these challenges, which are to integrate invasive clinical data that are coming from catheter mapping and are acquired during sinus rhythm in order to build a model, in this case of the left ventricle electrophysiology, in particular of the endocardial layer. And then we will try to see the predictive power of this model in a different setting, so using a pacing protocol in order to try to simulate ventricular tachycardia. So the data that we use is this set of signals that are acquired from a catheter, and this signal are usually post-processed in order to obtain these activation map, which are analyzed by the clinician and gives the initial information about the activation pattern in sinus rhythm. And we built a first pipeline in order to post-process this data in order to extract meaningful information, such as conduction velocities, which could be used in order to understand the presence of tissue, of scarring tissue or of gray zone in this layer, and can give some first quantitative information to the clinician, and that have been analyzed in this work. And then they could be useful also to parameterize the electrophysiological mode. So the idea is to properly choose, for example, the conductivity in tensors starting from this invasive data, in order to, first of all, reproduce the behavior in sinus rhythm, and then to try to perform the pacing protocol in order to stimulate ventricular tachycardia. And so this is the let's say the whole picture. And we have implemented this in our numerical code, and this is the results that we have obtained for one patient in sinus rhythm. So here we are only trying to reproduce the activation pattern. And in order to quantify our ability to do it, we have computed some error. So we have compared the data we have used to construct the model to the one obtained with the numerical simulation, and we had an error of about 2.5%. But in order to be more, let's say, robust, we decided not to compare with the same data that we use for constructing the model, but with another set of data which is coming from another set of signal, which is the unipolar signals, which are acquired by single electrodes inside the chambers. And upon this set of activation times, we recompute the error, and also in this case we found that that the model is able to reach the same level of accuracy. And so what we did in details is that we decided to parameterize the conductivity tensor starting from the conduction velocities, because it is well known from the literature that the conduction velocities depends directly from the conductivity tensor and the terms coming from the ionic model. And then, in order to close the model, we need also to find a way to parameterize the conductance of the ionic model. In this case, we choose the Tentasher model, and we started our so we started from data coming from the literature, and we decided to tackle these four species, and we have tried, and so we have modified only these four species inside the model. And then once we have built all these our numerical model, we have tried to perform a stimulation protocol. So the idea is to release a train of impulse that is likely to induce arrhythmias. And we were not satisfied by these very first results that we have obtained, because the results weren't coherent with the reentry circuit that was mapped for this patient. Here you can see that the clinical data show a nice figure of 8 reentry, which is not obtained by this model, and in particular by this parameterization of the model, which is more close to the form of a localized re-entry. So it's more similar to a fibrillation than to a tachycardia. So we decided to change a little bit how we parameterize the refractory properties of the tissue, so the ionic properties. And to do so, to do this, we decided to rely on the data coming from measurement on humans in the border zone. And so we on one side, we were satisfied by our capability of the model of reproducing the conduction velocity. But on the other, we weren't satisfied by the fact that the action potential duration is fixed for all patients, while it is clearly heterogeneous. And we have seen in this yesterday in some talks that there is also a clear heterogeneity in physiological patients. So we need, of course, it is possible that it is worse in the pathological case. So in order to better understand how to control the action potential duration, we decided to use a sensitivity analysis in order to understand which conductance of our ionic model are the ones that are most affecting the action potential duration. And to do so, we use the variance-based global sensitivity analysis, which is a tool that is providing some indices that are reported here, which describe the capability of these ionic species conductance to modify the action potential duration. So from this picture, it is clear that only 2 of them are really relevant to modify the action potential duration. And so we decided to consider, let's say, a minimal model where we modify only 2 of them, so only the most relevant, and we ran some simulation in order to see if there is a set of these conductants that is leading to a correct approximation of the ventricular tachycardia. And so this kind of behavior was also confirmed by this other sensitivity tool, which is the main effect plot. And so in the end, we decided to rely only on 2 of them. And so this is the result for the patient that we have seen before. So I just recall that, so the model it's built only starting from data from sinus rhythm, and we try to use this model to predict ventricular tachycardia using a classical stimulation protocol. And here we can see that we are able to get closer to the actual figure of 8 that is represented, that was measured in the patient. And we are not totally satisfied because there are some instabilities that still occur. So we need to, let's say, to modify the parameters in order to find an even better configuration. And we did the same for another patient. So also in this case, we have started from clinical data acquired in sinus rhythm, and we have tried to predict VT, and in this case, the blue and yellow line are the boundaries of the isthmus that were identified by the clinicians. And also in this case, we can see that we were able to reproduce a figure of 8 inside the channel. And also in this case, the reentry was not sustained, but it's we were able in this case to reproduce in a more accurate way the re-entry that was observed clinically. So so here are some reference. I'll thank you for the attention, for your attention, and if you have any question, I will be happy to answer.
Okay, thank you very much, Stefano, for the very clear presentation. And I'm going to open the question time. And if you have comments, please raise your hands. I see Professor Dossel hand, and again, is your turn, Professor.
Yes, thank you. It's me again. Sorry about that. I was very impressed about this talk. Thank you very much, Stefano. But one point I think is a bit strange for me. The conductivity tensor is very anisotropic. You very often pointed out that the conductivity is a tensor and not a scalar, which means that also the conduction velocity is very anisotropic. And if I remember right, we have made measurements on the anisotropy of the conduction velocity, and we found sometimes a factor of 3 from in fiber direction conduction velocity and orthogonal, perpendicular to fiber direction conduction velocity. And from my point of view, you can never pick up this with just measurements in the sinus rhythm. You need a second measurement with kind of orthogonal wavefront. Otherwise, you will never pick up this large anisotropy. And then I will, on the long run, see problems with the parameterization or personalization.
So thank you for the question, and I think that it's true. You need another source of let's say another source of information in the form of another pacing protocol in order to have a second map in order to infer also the let's say the information about the conductivity tensor in the direction in the cross direction to the fiber. And unfortunately, this is very rare, because we are talking about patients that are in the so we here the data are acquired during the procedure, so it's usually the clinician try to minimize the time of the mapping in order to avoid very long procedure. So what we did is that we still rely on the on a fiber structure, and so there is a rule-based model that we have fitted, and then we have projected them on the fiber, let's say the main velocity. So the conduction velocity was projected on these fibers in order to have a first estimation of which is the velocity along the fiber, the conductivity along the fiber, and the conductivity in the, for example, in the cross direction. And of course, I think that this is an approximation, and this is a choice. And I totally agree that the model would have a better accuracy if we consider another source of information. But I think that it is unlikely that this information will be acquired in the standard clinical intervention. So that's the difficult trade-off of building a model upon the available clinical data.
So thank you to both Professor Dossel and Stefano for the exchange of opinions. And there is time for a very quick question. I see the hand of Natalia raised, so please Go ahead, Natalia.
I was wondering whether you also have the MRI scan of the patient that you used, or whether, so we did this a study in which we calculated conduction velocity using clinical measurements, but also the MRI maps, because the MRI maps provide some sort of a, of course, you understand structural remodeling, and you can see you have another test of where conduction velocity potentially could be changed. So we correlated that with the MRI scans, and the study was published in Circulation, Arrhythmia, and Electrophysiology. And so it might be useful to, if you're doing this again from another patient data, to also have the MRI scan, because they're now required pretty much for anybody who's undergoing ablation. It can be another helpful tool to correlate, and might substitute what Olaf is talking about another data set that can help you sort of better determine what is the distribution of conduction velocity.
Yeah, totally agree with your point, and we haven't used data coming from images because it was not available for those patients that we have analyzed, but I think that it's let's say that it's for sure an important source of information that could be used, and I think that it is easily we can integrate in a easy way this in the model, and maybe trying to leverage information coming from mapping and from images could also help to avoid errors, that measurement errors that maybe are present in one of the two clinical data, and so I think that this will help to gain a better accuracy in the model, and maybe also to being able to reproduce the tachycardia, the ventricular tachycardia, in a more precise manner.